Hello, everyone. A few weeks ago, Mr. England had assigned DNA notes along with some key concept questions. Unfortunately, a majority of people have not been able to actually see that presentation. I am replacing that assignment with this presentation instead. There's going to be a different assignment assigned to this one. So if you've already done that assignment, that's great. This will be more of a review, and we'll also introduce some new concepts that Mr. England did not have a chance to go over in his presentation. So please watch the whole thing through and make sure that you're paying attention to the new math slides that are in here. So this is on the history and discovery of the structure and role of DNA. This picture here is very famous because this is Watson and Crick. They were the first to come up with a 3D model of what DNA looked like. And this is what you see in the picture here. So in 1869, which was only about 215 years ago, Friedrich Miescher was a Swiss chemist and was the first to identify DNA as a unique molecule. That means that he found it, but he didn't know what it was. He didn't know its function, and he didn't know how important it was going to be to the world of science. In 1914, Robert Fulgen found that all cells in an organism had the same amount of DNA, and that gametes, which are the egg and the sperm cells, had half the normal amount. But he didn't know why. In 1928, Robert Griffith demonstrated that bacteria could be transformed by adding a genetic factor from a deadly bacteria to a non-deadly bacteria, and it transformed it into a deadly bacteria. He didn't know what this genetic factor was. He didn't know that it was DNA that was causing this change. He just knew that there was something there that was causing this non-deadly bacteria to suddenly kill all the mice in his lab. In 1944, Avery, MacLeod, and McCarthy proved that DNA was responsible for the transformation of bacterial cells. Of course, in science, we can't do just one thing. If you had a chance to look at the Keystone Species and Trophic Cascade assignment, you'll see that in science, we have to repeat their experiment and change the conditions and see if it's the same across different areas, basically. In 1952, Alfred Hershey and Martha Chase demonstrated that DNA is the genetic factor that Robert Griffith was looking for, not proteins. At the time, they just thought it was proteins. So, in the 1940s, Erwin Chargaff was another scientist who was able to determine a few things about nucleotides in DNA. Based on the observations, two rules can be deduced. In purple, you see adenine and guanine, A and G. When you add them together, they equal 50% of the nucleotides. And when you add thymine and cytosine, the T and the C, they also equal 50% of the nucleotides. The other thing that you notice about Chargaff's rule is that the percentages of nucleotides, the amounts of nucleotides in each organism are very different for each species. Here we have humans, wheat, a bacteria, and a virus. As you can see, they all have different amounts of these four nucleotides. In 1951, Rosalind Franklin used an X-ray crystallography machine to determine that DNA was double-stranded, a helix shape, and that phosphates were on the outside of the ladder. Her work did not include base pairings. Chargaff concentrated on nucleotides, meaning the inner part of DNA. Rosalind Franklin was working on the outside of DNA. This picture here, with the X in the middle, those dots are the bases. The dark bands on the outside are the sugar phosphate backbone. So the DNA off to the right that you see, the direction of the arrow is the direction in which she was taking the picture. So she was looking at the DNA from, quote unquote, the top. So now we come to James Watson and Francis Crick. James Watson and Francis Crick determined the structure of DNA by making models. They were able to determine that the sugar and phosphates were on the outside and also that the bases formed the rungs of the ladder. When we say ladder, we mean that if we took the DNA helix and we untwisted it and flattened it, it would look like a ladder. Now they needed to figure out how the bases fit together inside this ladder. So using Franklin's X-ray crystallography, Watson and Crick found that the bonding, that A and G are purines, and they're too wide to be paired together. They would end up overlapping and causing a lot of space issues. Two pyrimidines, T and C, are too far apart to form the hydrogen bonds that needed to stay together. But they found that a purine and a pyrimidine, so one big base and one small base, fit perfectly in their measurements. So at the time, Rosalind Franklin was working with a man named Maurice Wilkins. It was his lab, and at the time, women were not allowed to really be able to function on their own as scientists. If they did, their work wasn't taken very seriously. Well, Wilkins shared Franklin's information, her data from her crystallography, without her permission, and he shared it with Watson and Crick. So when Watson and Crick were working on their model, they used Rosalind Franklin's and Erwin Chargaff's information that they gathered and created their model. Once they did that, Watson, Crick, and Wilkins applied for the Nobel Prize, and they won. So Franklin did not get the recognition before she died. She was not allowed to be awarded the Nobel Prize after she had died. It's just not possible because the Nobel Prize is about $1 million as a cash prize. And only up to 3 people can be nominated for the Nobel Prize. So Watson, Crick, and Wilkins won the Nobel Prize. Chargaff also did not get any credit or a share of the Nobel Prize money. So up until recently, Rosalind Franklin did not get the recognition that she deserved for her part in leading up to the DNA model that Watson and Crick created. And this was only in 1962. So if you think about it, it's really not that long ago in history. So the next slides you're going to be using to complete your Go Formative assignment. It's going to be titled Nitrogen Base Percentage Practice. And this will be important information for your quiz later on as well. So this part of it, at least, you will want to make sure you have in a safe place, like notes or something like that, so that you can come back to it when we start going through our study guide. So we have Chargaff's rule. We know that when the 2 bigger bases, A and G, are added together, we get 50% of our nucleotides. When we have T and C together, we have the other 50% of our nucleotides. Watson and Crick determined that means, based on the pairings, it has to be 1 big base and 1 little base, that the amount of adenine and thymine are the same, and the amounts of guanine and cytosine are the same. So here's our example problem. If we have 10% of adenine, how much do we have of thymine, guanine, and cytosine? So we know that A equals T. We know that 10% of adenine means we have 10% of thymine. So we have 2 of our 4 bases covered. In step 2, you add those 2 amounts together. So 10% of adenine and 10% of thymine means that we have 20% of adenine thymine total. When we're talking about percents, the most that you can have is 100. So you have 100 minus the 20% adenine thymine, and that will give us the rest of guanine and cytosine. So we have 80% of the guanine cytosine pairing. We know that guanine and cytosine, they equal each other, just the same way that adenine and thymine equal each other. So we have to divide that amount by 2. So 80% of guanine and cytosine divided by 2 is 40% guanine and 40% cytosine. So our final amounts would be our original 10% adenine, 10% thymine, 40% guanine, and 40% cytosine. An easy way to double check to make sure that you have all the right amounts are adenine and thymine the same, are guanine and cytosine the same, and do all four equal 100%. Okay. So now here's the structure of our nucleotide. On the left, in the yellow boxes, we have our purines, which are our longer bases, our adenine and guanine, A and G. As you can see, they have two sections to them. That's why they're the longer bases. On the right side, we have the pyrimidines, which are the smaller bases. Here's where we discuss how different DNA and RNA is. We have cytosine and thymine in DNA, so A and T in DNA. But in RNA, there are no thymines. Instead, we have our uracils. So in RNA, instead of a T, it's a U, so AU in RNA. The entire nucleotide itself is made of three pieces. Our pentose sugar, as you can see in blue, we have our deoxyribose, which is where DNA gets its name from. And then we have ribose, which is where we get our RNA from. The next part is our base. So it can be adenine, guanine, cytosine, thymine, or uracil. And the last piece is the phosphate group, as you can see in the bottom left-hand corner. Forming the double helix. So here you see three different ways that people show the structure of DNA. To our left, it clearly shows the differences in the bases. So you can see where C and G are together and A and T are. Our middle structure shows our ladder after we've taken our DNA helix and flattened it. We can see now that A and T, they have two hydrogen bonds to keep them together. And G and C, we have three bonds to keep them together. That's how we know that they match. They're like puzzle pieces that come together. Our picture on the right is a space filling model. This is just an idea of how it looks like in a 3D model. Unfortunately, it is very difficult to take a picture of DNA because it is so small that we have to use artist's interpretation, which gives us a pretty good visual, but it's hard for the general public to know what the actual structure of DNA looks like because we don't usually see a picture with our own eyes. DNA forming. So structures in eukaryotes, the DNA is wrapped around proteins called histones. So in this top picture, you see here that the red line is our DNA helix. And these green pieces here are the histone protein, and they keep wrapping and wrapping around it. This structure right here, the DNA and the histones together, this is called a nucleosome. Now, on this bottom picture here, we see that the DNA helix is winding and getting tighter and tighter and tighter until it becomes these cords almost. Then those wrap around these histones, and they keep wrapping and wrapping and wrapping, and they keep going and keep going, and turning into thicker cords, because they're continuously wrapping around themselves too, into a chromatin fiber. The chromatin continues to coil and twist until it reaches its final structure of the chromosome. The only time that you will see this X chromosome structure is during the cell cycle during mitosis. when the cell is getting ready to split into two. Outside of that, it's usually loosened in this chromatin form. So that is all of the important information that you guys need to know about DNA. You have your nitrogen base percentage worksheet to do on GoFormative. If you have any questions, please feel free to ask, and keep this set of notes, this video in general, ready to be watched again. It will stay in Edpuzzle for as long as you need it. You're welcome to watch it as many times as you need it. And hopefully it will help you when we get closer to the quiz and with your studying.